All right, good afternoon, everybody. We're here to go ahead and cover section 2.4, chemical reactions and enzymes. As you came out of section 3, the four macromolecules that are required for all living things are carbohydrates, nucleic acids, lipids, and proteins. One of those specific functions of proteins was enzymes. However, everything within a living organism has to happen because of a chemical reaction. Chemical reactions are just a process by which one set of compounds or chemicals is changed into another. For example, you could have a slow reaction like the rusting of this bolt, which is the combination of iron and oxygen over a period of time, and the product coming out is iron oxide or rust. Or you could have a very fast chemical reaction, such as when the Hindenburg, which was filled with hydrogen gas, ignited in the presence of oxygen. That resulted in a very violent or fast chemical reaction, and very explosive and giving off lots of energy. In the reaction below, let's go ahead and identify the reactants and the products. So carbon dioxide and water, carbon dioxide plus water, and then we have this arrow which represents yields or produces glucose and oxygen. So carbon dioxide and water in this chemical reaction are the two reactants. The two products coming out are glucose and oxygen. Chemical reactions involve the breaking of chemical bonds in the ingredients or the reactants and the formation of new bonds in the products. So the bonds that are holding the CO2 and the water together during photosynthesis in the presence of light, the bonds are being broken, the atoms are being rearranged, and new bonds are being formed to produce some different products. Chemical reactions that release energy often occur spontaneously or kind of on their own. And these chemical reactions that absorb energy often require a source of energy to get them started. So to kind of navigate through a chemical reaction, we have to understand what activation energy is. Activation energy is the amount of energy that is required to start the chemical reaction. So whether the overall chemical reaction releases or absorbs energy really kind of depends on how much activation energy is needed. All chemical reactions require some input, some activation energy. And some require more and some require a little bit less. Whenever a chemical bond in a compound is formed, energy is being stored in that bond. If a chemical bond is being broken, energy is released. So you have two graphics on your outline there. And if you look at the course of the reaction, kind of flowing from left to right, we have reactants and we have products in each one of these. And we have the y-axis labeled with the energy input. In the reaction that's energy absorbing, this one on the left, the reactants are starting at a lower energy level. A certain amount of activation energy is required to get the reaction started. Once here at the top, the reaction will take place and some products are being produced. So our products coming out have energy stored within them. So they're coming out at a higher energy level. In an energy releasing reaction, the reactants start at a higher energy level. The amount of activation energy is required to get the reaction to take place. Once here, the reaction will actually occur and the products will come out at the end. Now, energy is being given off in this case, meaning the products that are formed are at a much lower energy state. This is significant for living things in the sense that every organism must have a source of energy to carry out its own chemical reactions. So how does this define within a living cell? Well, enzymes are what are known as biological catalysts. And these are substances that will speed up the rate at which a chemical reaction will actually take place. And they do so, enzymes lower the activation energy, which can then speed up the reaction. So if you look at the graph over here on the right-hand side, you have two different pathways. The blue pathway would be following the chemical reaction without the use of an enzyme, and the red is if an enzyme was used. And you'll notice the amount of activation energy is a lot less. It's a lot lower. So imagine you had a cart and you were pushing it up the hill. It would take a lot less energy to push it to the top of this hill to get the reaction to take place versus, the blue curve here. So enzymes speed up this reaction by lowering the activation energy. So in terms of enzymes and their involvement in chemical reactions, how many reactions does an enzyme catalyze? Well, they only catalyze one. They're very specific. So one enzyme will only speed up one particular chemical reaction. And the name for the enzyme is often derived or come up from the name of the reaction that it catalyzes. For example, the disaccharide maltose, this is a double sugar, in combination with the enzyme maltase, will be broken down to produce glucose, 2 glucose molecules, and the enzyme. So the enzyme maltase binds to only one particular substance, and that in this case is maltose. It's very specific. And it breaks that double sugar down into its two components, glucose molecules. Enzymes, as their structure holds, they have a site where they can bind to the reactants. So for example, maltase has what is known as an active site where it will connect and bond to the sugar, maltose, in order to carry out the chemical reaction. These sites are referred to as the active site. So if we look at the reaction as a whole, the substance that an enzyme works to catalyze is referred to as a substrate. So these are any reactants in one of those chemical reactions. The active site is the location on the enzyme where the substrate and the enzyme bind together. Once that happens, they form this large kind of complex known as the enzyme substrate complex. And oftentimes the enzymes are described fitting with the substrate like a lock and a key. That one enzyme has an active site that is unique, where only one substrate can actually fit within it. Once that enzyme and substrate are sort of bonded to one another at the active site, the chemical reaction will take place, and then the products will be released. The good thing about enzymes and how they speed up reactions is that they're never used up during the chemical reaction. They can be used over and over. So this enzyme can react with more substrate, carry out the chemical reaction, release whatever products are coming out of that, and then react with more substrate. And they will continue to be recycled and reused, as long as this active site isn't changed in any way. So enzymes function to speed up the rate of chemical reactions. They're lowering the activation energy. They're bonding to substrates and helping to either break down or build up and produce some sort of product in the end. Now, enzymes, like any other compound, are sensitive to their structure. So enzymes are made of proteins, and they are affected by changes in temperature. For example, there's certain enzymes in your body that will work best around, your body temperature of 37 degrees Celsius. If your body temperature goes beyond that range, like for example, when you're sick and you have a fever, the higher that body temperature goes, that impacts how these enzymes actually function. So running a high fever that is not brought down can actually kill you. So it alters how those enzymes function. So temperature, enzymes are very sensitive to temperature changes. Enzymes are also sensitive to changes in pH. They tend to work best at certain pH values. If you studied, pH a little bit in junior high, there's a pH scale from 1 to 14, where acidic substances have a pH from 1 to less than 7. 7 is considered neutral, and then anything above 7 is considered basic. So in your stomach, in your digestive system, you got a variety of enzymes that help to break down carbohydrates and proteins and fats that you consume. In your stomach, in particular, there's enzymes that are activated in the stomach only because the stomach has an acidic pH value. Outside of the stomach, these enzymes don't function at all. So you consume proteins, you're going to digest the protein in your stomach with these acid functioning enzymes, and the proteins are broken down. But once those stomach contents leave your stomach and enter your small intestine, those enzymes no longer function. They only work well in that acidic environment. When enzymes get pushed outside of their normal functioning range, whether that be extreme temperatures, cold or hot, or their pH value, the structure of the enzyme itself changes, which means the protein structure is changed. And we call that denaturing. We say that the enzyme is denatured. So what's happening is the protein that makes up the shape of that bonding site to the substrate really gets altered, and the enzyme can no longer bind to that particular substrate, and the chemical reaction will cease to occur. Now, as we progress later in the week, we're actually going to perform some enzyme experiments and give you a visual demonstration of how enzymes function in carrying out chemical reactions, but also how they can be influenced by outside factors as well.